Hello, everyone, and welcome to today's live broadcast, Innovation in Lab Automation, the Future of Computational Biology. It is presented by Mark Bünger, an analyst at Innovation Lab. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located on the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Mark Bünger. I will now turn the presentation over to him.
Okay, thank you very much. Innovation and lab automation is a really fascinating field, and the reason it's a computational biology enabler and the future of that is because so much of what we know in computational biology today isn't directly accessible to our senses. It's not easy to do in a replicable manner, and our tools are really, really critical to how this field advances. At the same time, advances in the field have been bringing up new things to measure and new important phenomena that have driven the evolution of the tools themselves. So there's a lot going on. If I were to break this down, what's driving innovation in lab automation, it really comes down to a few enablers. So one is just technological advances, things like miniaturization and integration that we see happening in a lot of types of technologies, sensors, microprocessors, motor control, and so on. There are also or sort of approaches to innovation, like open innovation and open source. These are important because they're basically helping us spread knowledge and understanding of not just the science itself, but also how to make the tools and how to use the tools. And by basically getting on the same page on a lot of these fields, because we can spread information about them so much more quickly, we really become essentially more of a global integrated team and global collaborators in a way that we haven't been able to be in the past. And then there's also new business models. So these are ways that we can get information into users' hands and into new fields, like spinning out companies from universities or corporations by creating startups and funding them with crowdsourced money or with venture capital, and then accelerators, a new type of business that essentially takes startups and helps them get over the really mundane hurdles that have kept a lot of startups from getting to market in the past. Just basic things like legal services and so on, but also ideas about how to design things to be manufactured at scale, how to market a business, and how to sell a product that inventors often don't have in their skill set. So all of these enablers on the one side, as they've been evolving from the past to the future, have been really driving some results in lab automation that we can see in a couple different stages. So first, it was really just about lower cost and better R&D productivity. Lab automation was a way to automate the work of very expensive, highly educated, highly paid researchers, and essentially offload some of the tasks that they're having to do. That was, pipetting or recording information in a lab notebook, just basic tasks that, most industries, whether they be banking or manufacturing or music, all these industries have benefited from automation, and R&D is really no different. The second thing, greater accessibility and the democratization of science, that's really related to what I was saying earlier about open innovation and open source. While those are really terms that come from another field, the move towards open science is also very important and a catalyst that works with those other two factors. And basically, it's bringing a lot more people into the scientific field. More scientists alive today, I think, in all of history combined, a stat I've heard, so must be true. Heard it on the internet, I think. But at any rate, there are a lot more people doing science today. The ability to do science has been expanded considerably. Where we are right now, I'd say the current state of the art is around consistency. So in a lot of scientific fields, and in biology in particular, there's a crisis of reproducibility. And basically what that means is that two people doing the same experiment, following the same protocol, don't always get the same results. Some of it inherent in the nature of biology, and some of it's just because of avoidable factors and automation and the consistency that the data and robotics and other types of automation tools can bring really helps reduce some of that inconsistency. And then what's next, or the future, I think what we're seeing is basically scientific frontiers. In other words, completely new measurements and calculations, things that we can't really measure today or don't understand the impact of. And towards the end of this presentation, I'll touch on one really interesting future area, which is quantum computing, and the ability it might give us to do modeling and analysis of biological data and lab data in a much more rapid fashion, and use bigger data sets and lots of other things. So if those are the enablers and the results driving innovation and lab automation, well, why is it happening? Like, what's the specific thing that's driving it? And it's pretty simple. It's actually what's happening in a lot of other fields. Lab tools are just following office tools and recording instruments for music, media, and a lot of other things. They're basically becoming information technology. And so if you go back to the 80s, when we started getting desktop computers, really were literally meant to replace typewriters. They were office tools. And that's why companies like IBM and Xerox that were in the space put a lot of effort into becoming information technology companies. And while there's an obvious component to of information to science, we haven't always thought about scientific tools as information technology. We think of them as scientific instruments. We think of the processes as something that's, certainly about discovery, but the rise of omics, so genomics, and informatics, so bioinformatics, for example, that's really transforming science and scientific tools into an information technology space. So if you look at something like Moore's law, the experience curve about the increasing performance of chips and computers, that's kind of become synonymous with rapid exponential improvement in anything. So we'll talk about Moore's law in cars or things that really have nothing at all to do with the original law itself. And that's actually true in a lot of other fields. So as we've looked at, IT startups breaking more and more industries and making new ones, again, we can go through journalism and retail and a bunch of other areas. That's really what's happening, starting to happen in the scientific field, and for the same reasons. So it's basically the use of information, automation, and open source that has driven this in software and in electronic hardware, and those two things have pervaded all kinds of fields. So things that used to not have a software component, like, again, let's say the music recording industry, as those became digitized in a lot of ways, that really transformed the industry and made something different out of it and brought new people into playing it. That's what's happening right now in science, and that's one of the reasons we see a lot of science hacker groups, so DIY Bio, for example, and others where people that have certainly a technical background, but don't always have access to all the tools that you normally get in a big lab at a university or a company, are doing really, really interesting scientific innovation and making some breakthroughs. At the same time, people that do have access to those tools are saying, well, why are we spending so much money, and why are we basically having what's becoming quickly obsolete technology when there is a field of innovation that's bringing a lot more things to bear? So the innovation that's coming out of all of these fields, again, from office equipment to now more scientific instrumentation and lab automation, has two big impacts. One is speed to market, not only for the tools themselves, but for what people are making. So it's this ability to come up with a hypothesis, test it, iterate, revise, and test it again, that's driving speed to market. In other words, that does things that process take weeks or months or even years, and now it can take hours or days. The other thing that's really remarkable is the breadth of products. So in the past, it used to be very much, what you started looking at in terms of, say, a medical molecule or something that was for agricultural biotech was the only place you'd find a product or an application of that research. And today, more and more, these tools are providing what you would call a platform ability. In other words, you develop something that then can create lots and lots of different kinds of products. So companies like Ginkgo Bioworks and Amyris do everything from fuels to agricultural chemicals to flavors and scents. That really fundamental platform ability also spreads to a lot of other startups in not just outside of biology, but in it. So we're looking at now consumer products, foods, personal care products, and things like that we never would have thought of as biological, rather biotech. But because these tools are so accessible and so applicable in a wide range of fields and products, it's really expanding the scope of what can be addressed with biotechnology. So as these things are entering labs, as I said earlier, we often use Moore's law as a metaphor for something changing really, really fast. But in fact, biological technologies and the instruments for creating them are evolving even more quickly. And so the equivalent of Moore's law for biotech is called Carlson curves. Rob Carlson started noticing this pattern in specifically in commercially available DNA sequencing and synthesis instruments. And so he started basically plotting the productivity of those tools, both in terms of time and cost, going back to the 70s when a lot of them were invented, up to the Human Genome Project, and then today where we have dozens of companies entering these fields. In fact, the pace of improvement in those instruments is faster than what we're seeing in chips. So it's actually using Moore's law. And there are two, broadly speaking, two aspects of this. So that's not just automation. In other words, it's not just a tool that moves faster. It's also information. Our ability to capture and analyze information is expanding. And when we can do that in a rigorous way with these tools, we can also share and spread them, which again, which brings more people to the table and helps us to process a lot more things in parallel. The automation also helps in that it reduces not only the time, but also the skill needed to do a lot of biological tasks. So in the past, it would be, you would need to spend maybe, months, maybe years learning to perfect a certain technique just on the ability to use your hands or the ability to identify markers in a sample using, just using your eyes. And the tools are getting much better at actually using machine vision or robotics to automate physical and mental tasks so that we can use tools to see patterns that we really needed to learn to do on our own over a long time. So break, the lab of technology, the lab of tomorrow and its technologies down into a few categories and some specific things that we can start to look at. This isn't exhaustive, but I think it's actually a pretty comprehensive picture of the areas that are important to be looking at today. So on the information side, there's BioCAD, computer-aided design, and cloud BioLIMS, so laboratory information management systems. Those are the two really information technology, software types of things that are driving this. And then on the automation, a number of things, again, physical things like DNA synthesis and CRISPR and so on. But for today's talk, we're talking about lab automation. I just want to focus on open source lab equipment. Advances in a lot of different forms of lab equipment, of course, but open source, I think, is really the area where we see the fastest and most widespread breakthroughs. And I'll go into each of these in turn. So on BioCAD, so CAD stands for computer-aided design, and CAD actually started in things like architecture and mechanical types of engineering. But more recently, it's come into biology. And so we can take the same types of tools where we have an in silico that is in computer model of a process or of an object, and that model corresponds, its behavior in the computer corresponds accurately enough to its behavior in the real world that we can start to do things like simulation, iteration, permutation online or again in the computer, so that we don't have to do it in the real world. So the same way we don't have to crash test cars from the first prototype, we can develop prototypes, and then when we are pretty sure we've figured out all the biggest problems, we can actually crash test physical cars. We can do the same thing with BioCAD. In other words, we use these tools to automate the design of, let's say, a gene, and we can model its expression and the results of any mutations we might make, and get a pretty good picture of how that's going to work a lot more quickly than we could do that if we were to actually put that gene into a microorganism and then try and culture it and see what the results were. So not only does it automate engineering work and identify errors early on, errors in our reasoning or errors in our process, it can also help with permutation. So again, let's say that you have an idea about one mutation and how that might play out in a biological system. With BioCAD systems, you can actually have it generate mutations that you either might not have had the time to or might not have had the knowledge to do, and then run those systems also through a virtual simulation. So those things really help, again, accelerate the development of biological organisms and tools. And there are, in addition to the tools themselves, a couple of really interesting integrations we've seen. So doing modeling on its own has some, it's limited. In other words, it's one step in what is always a much longer process. So we're seeing companies integrate that ability with other steps in the process. So Atom, which used to be called DNA 2.0, started actually synthesizing genes, and they developed some software, CAD front end, basically, to be their customer interface. So it's kind of like, if you go buy a car, you might figure it online first and then you order it. That's really similar to what they've done. They've developed tools so that you can test out what you think is going to work in DNA on a virtual environment, and then when you've got your candidates chosen, you can actually order them, and they'll send you the actual plasmids with those sequences that you've discovered. Another example is benchling. So benchling also makes some of this type of design software, but what they've looked at is the increasing reliance on collaboration. So in the past, again, a lot of people were sort of worked on their own or didn't necessarily have the ability to easily share work with anybody outside of their specific lab. But now with collaboration becoming so widespread, across an institution or even across the world, the ability to integrate collaborative tools with every task is important. So they've been looking at that. Moving on to biomems, that again is laboratory information management systems. Electronic lab notebooks are one aspect of that, but those systems have actually been around for quite a long time. But two changes, the bio and the cloud, have really helped accelerate their usefulness and their adoption. So again, in the past, laboratory information systems were used for things like, keeping track of how many reagents were, how much various reagents were left in inventory, or who is doing what experiments on what machine at a certain time. So scheduling and pretty mundane types of things. The addition of biological to that essentially meant that these systems were customized for biological data and often integrated with the instruments that were capturing it. So rather than having to look at a display, write it in a lab notebook, and then go type it up in the LIMS system, all that data flow directly from the instrument into this LIMS system is automated these days, and again, adjusted or customized for bio. Cloud basically means that it's in a shared system from the beginning. So as improvements to the software are made, they're instantly available to all the users. And similarly, as data is acquired, it's also available to everybody who has access to that data through the cloud system. So these changes have, again, happened in a lot of other areas. Think of banking and other types of industries that have been doing this for quite a while. The fact that it's coming to into lab automation could have similar impacts where, we have a lot more automated back office and front office work in those industries, and that could similarly affect how we do biology. In addition to just the workflow improvements, it also has the benefit that we can all get access to faster processors and a lot more data storage, so high-end tools, D-level, and again, some things that are just kind of practical aspects of what's happened in IT and a lot of other industries for a long time. My personal view, though, and one that a lot of people would share, is that the real critical aspect of this that's specific and unique to biotechnology is the crisis of irreproducible results. And that's basically because a lot of times we'll have, as I mentioned earlier, we'll have a protocol that's published in a paper. You'll read that paper and you'll try and follow it, and you just don't get the same results. And it's really frustrating and hard to understand exactly why. There are a lot of reasons for this, and some of it is, again, just due to experimental noise, the inherent nature of biology. Every organism is a little bit different from every other one, and sometimes those differences actually matter. Sometimes it's also just a difference in technique or a lack of information about, for example, what temperature something was stored at, or other conditions that were maybe just seemed like irrelevant details to the person who originally did the experiment, but are actually very important now and affect the outcome of the experiment. So those factors are greatly aided by BioLim systems, and two companies that are looking at this very directly, Calvert and Riffin, and they I think do a huge service by starting with design of experiments, DOE, and helping researchers actually document how they're going to do experiments and set them up in advance of running them. So that helps them run the experiment in a more logical way, but it also helps with documentation. There are also a number of companies that are working on LIMS integrated robots. So the Emerald, Synthego, and a few of those. But here again, the ability to use the software to guide the hardware, the robotics, is really critical, because you eliminate a lot of that error that results from one researcher versus another actually doing the experiment. And the robots, in the best case, are actually going to do it consistently, not only every single time in a certain lab, but also from one lab to another. So open source, there's a lot of flavors. So there's open source software, open source hardware, and we're seeing increasingly open source science. There are a lot of areas of science where it's better to have a large number of sources of data, and so open source science is basically helping address that need. The designs then for open source science and that idea of sharing is also coming to hardware and software outside, on the edges of science. So Arduino microcontrollers, RepRap 3D printers, CubeSats. This is an open standard for designing satellites, and now we have a bunch of very inexpensive satellites in orbit using that standard. Lower the cost and expand the user base, and that basically helps improve the tool's performance and cost further as more people come in. So Chido and Opentrons are two examples of automation in PCR and pipetting, respectively. But a lot of companies that are bringing instruments to market are now taking this approach. Essentially, it has a lot of advantages in terms of getting to market and getting products into people's hands at a lower cost and more broadly. So I mentioned earlier quantum computing, and I want to say, I would love to say it's next. It's not quite next, and so I want to give a little overview of what quantum computing is and when I think it'll actually be available, based on not only what you can read online and in some papers, but also what I've learned talking with the experts on both sides, both the user and the developer side of quantum machines. So the basic thing to know about quantum computing is that whereas normal computers use bits, so 1 or a 0, quantum computers use qubits. So if you think of this sphere over here, which is a way that qubits are often depicted, normally you would have a 1, say, an arrow pointing straight north, or a 0, the arrow pointing straight south. Qubits actually give you a superposition of all of the possible directions of that arrow, which corresponds to basically different probabilities between that 1 and that 0. And what that means basically in practical terms is that whereas a normal computer, a conventional computer, would have to crunch through all the permutations of 1s and 0s one by one, a quantum computer, because of the substrate that it uses for calculation, can actually look at all of those possibilities at the same time. So that's a really far too brief introduction. I encourage you to look into the basics of quantum computing if you want to know more. But the implication of that is we could do very complex calculations very quickly. And so in chemistry, obviously, chemistry and biochemistry, there have been a lot of papers and even Nature and other high-profile publications talking about how quickly quantum computing is going to be solving these chemistry problems. Richard Feynman also said nature is quantum, and if we're going to solve it, then we're going to need computers. So the reality, though, again, just based on conversations recently with the developers and the users of these, is that it's quite a long way from being that. It's even modeling, say, a 3-atom molecule is kind of at the edge of what's even conceivable now with today's quantum computers. And with other computers, that could take off in the future, but it will be still going at least for the next few years, where 3 or 4 atom molecules might be the most complex things that we can model. But after that, hopefully it will take off, and maybe in the 25 to 2030 timeframe, we'll have some amazing new tools. For most of us, that's a little bit farther out than the horizon we need to normally consider. So if you are in lab instrumentation and computation, then yes, you should be computing and how it could impact your business. But if you're as a user, it's definitely worth it to wait a few years for about how that could be your science. So to wrap this all up then, how can you get started? If these are areas that you're interested in, I would say look at investors, breakout labs with the Bill and Melinda Gates Foundation. Both of these are funded by IT billionaires, but they're really trying out some of the most interesting new things in automation, both in tools and in using them. There are accelerators like the QB3 Garage at UCSF, Y Combinator, which is a very famous IT accelerator, has recently added a biotech track. IndieBio, Lumina accelerators, there's a lot more focus on those things. There are hacker spaces like GenSpace and BioCurious and Counterculture Labs in Oakland. There's the IGEM, that's Internationally Genetically Engineered Machines Competition. Crowdfunding sources, Indiegogo has been pretty open to biological both tools and organisms, whereas Kickstarter actually had some biological organisms that they said we're not doing that again. But there is some innovation happening there. And then there are events. So I mentioned IGEM, that's actually a competition, not a hackerspace per se, but it's a competition for students. And then there's an event called SynBioBeta that's will be held in San Francisco later this year in early October. And this will bring together a lot of the community of both the hardware developers and the users for these technologies. So there's a lot going on, and the most immediate way to get started is to send questions. So we have time for a few questions here today, and you can also follow with me either through the LabRoots tool or directly on email. Thank you very much.
Thank you, Mark, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click on the Send button. We'll answer as many of your questions as we have time for. So let's get started. First question is, there are a lot of little companies in this space. How will they compete with the Agilents and Thermo Fishers of the world?
Yeah, that's a really good question. And it's true, a lot of these companies are really scrappy and small, and they don't look like sort of the Goliath the David that would take on a Goliath. But what happens over time is these tools get better and improve, and it's very similar, I think, to what's happened in other industries where a low-cost competitor initially starts out only being applicable for amateurs and students, but over time it improves and really becomes the industry standard, not only because it improves, but also because the people that have been using it become more and more senior and more adept at using them. But another way is just through acquisition. So these tools often really, again, start out as small companies, and small companies get acquired. So we've seen this in a lot of different areas with Life Technologies and Agilent and others actually acquiring some of the software and hardware that other people have developed.
Great, thank you. What companies or industries are adopting this approach today?
One of the things I think is really interesting, I alluded to this in the conversation, is that sometimes it's definitely companies you'd expect, like in say pharma or in ag tech, but these tools have really broadened the appeal and the relevance of biologically based materials into industries that really wouldn't have had anything to do with it in the past. So one example from about 10 years ago is fuels and energy. The global warming and the war in Iraq and a bunch of other things that were happening about 10 years ago, and the oil price also at the time spurred a lot of interest in using biologically sourced materials and biological processes to create fuels, and then also other types of substitutes for petroleum products. So again, back then, with the oil price being well north of $100 a barrel, people developed biological ways to make really almost anything we would get from petroleum at costs that were competitive at that price. So oil prices have been creeping up again, and the opportunity to do those things might come back. But in the meantime, there are other things that are actually much more high value products that I wouldn't have expected to see as biological back then. And those, again, can be things like food and personal care products, things that are very intimately close to our bodies, and that a regulatory pathway that hasn't conventionally been geared up for biotech. But that's exactly what's happening right now, is that people are starting to look at how can we regulate and approve these products as they move to market, and that's happening more quickly. A lot of that is driven by the economics, but a lot of it's also by the people's shifting desires. In other words, wanting to have more natural products and wanting to use more biologically based things than those from other more synthetic chemical means.
Thank you for that. And for one more question. Besides quantum computing, what looks exciting about the future?
I think the future by definition is exciting. So I would say one area that I think is a great, I should say one trend I'm seeing is the use of artists. So artists in residence. There are a number of institutions and organizations that we would normally think of as very, very scientific that have brought in artists to help them see new possibilities with the work and the tools that they're developing. So outside of bio, SETI, the Search for Extraterrestrial Intelligence, has an artist in residence program. There's a guy named Scott Kildall there, who's basically been working with their data and their tools to expand their understanding of what they've got. But within biotech, Ginkgo Bioworks and a company, Modern Meadow, have basically been using artists to help them explore, what new products and services they might do in the future, how people might react to those, look at how they might be designed to be more appealing. There's an artist named Lucy McCrae who, while she's not a biological artist herself, she's been using sort of the ideas and tools of biology to look at things like, what if you could swallow a pill that would help you make your own perfume, or, what if you could essentially grow the things that your body needed specifically at a certain time based on your own cells. So these concepts, they can be very abstract, but then when an artist gets them and sort of puts them into, just envisions them basically, and does a little bit of the work, not only about thinking, but developing them to make them happen, it seems a lot more tangible. And we've seen, many, many times in the past where the tricorder on Star Trek has sort of inspired the first mobile phones and things like that. So I think looking to art as a way to see the future before other people do is a really good approach.
Very interesting. Thank you. I would like to once again thank Mark Bünger for his presentation. I'd also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through August 31st, 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Bye.